Hi, everyone, and welcome to the November meeting of the Linnaean Society of New York. I'm Debbie Mullins, the president, and I call the meeting to order. Thank you for tuning in tonight to hear Dr. Kelly Zamudio's presentation on neotropical frogs. With winter birds starting to appear in the area, we have several field trips coming up that you may be interested in. These include Rye Playland, led by Tom Burke and Gail Benson, Jones Beach and Point Lookout, led by Joe DiCostanzo, and there are still a few spots left on our February trip to the Adirondacks that will be led by Joan Collins. Please remember that members have priority over non-members for all our field trips. So if you're not a member and you're interested in going, please join the society. Visit our website, linnaeannewyork.org, and click on Members to find the membership application. You can also see a list of all our field trips through August of 2025 on the website. Well, the 125th annual Christmas bird count is coming up soon, and registration for the December 15th New Jersey Lower Hudson Count Circle that includes Manhattan is now open on the New York City Bird Alliance website. They also have a list of the contact people for other area counts, including Bronx and Westchester, Queens, Brooklyn, and Staten Island. And if you live in another part of the state, the dates and contact people for the other CBCs across the state can be found on the NYSOA website. That's New York State Ornithological Association. And for other areas, if you're outside of New York, please contact your local Audubon Society to find out when you can participate in their Christmas bird counts. I need to remind you that if you haven't paid your dues yet, please do so right away. This year, we seem to have an unusually large number of members who are dragging their heels on renewing their membership. So please take care of this as soon as possible. This fall, the board of directors has taken steps to help make it easier for people to join the Linnaean Society. For years, we've been hearing from people who told us that they would have joined earlier, but they didn't know anyone to sponsor them. Others have wondered who we're trying to keep out of the society by requiring a sponsor and having the membership vote to admit new members. In October, we eliminated the requirement for sponsorship, and just last week, the board of directors expressed approval for dropping the requirement that new members be voted in by the board and by the membership. We hope these changes send a clear signal that we would love for everyone with an interest in birds and natural history to become a member of the society. If you'd like to share your opinion about dropping the voting requirement, you can email me at president@linnaeannewyork.org. Last week, you were asked to vote on two motions, one to approve the minutes of the October meeting, and the second to approve the membership application of 16 new members. The minutes were approved with a vote of 141 in favor, none opposed, and 6 abstaining. And the new member applications were approved with 146 in favor, none opposed, and 1 abstaining. And this should be the last time that you will be asked to vote to accept new members. So please join me in welcoming these 16 new members: Melissa Breyer, Cheryl Craddock, Michelle DeMello, Rosemary Field, George Giannakos, Craig Himmelwright, Richard Lufrano, David Manoli, Sahar Segal, Anna Tom, Adrienne White, Julia Ding, Lisa Wolf, Thomas Friedman, Sarah Kimball Stevenson, and Erasmus Van Niekerk. Welcome to all of you. We're happy to have you as members of the Linnaean Society, and I look forward to meeting you in person on an upcoming field trip or at an event. So that concludes my announcements for tonight. As usual, we have muted your microphone and disabled the chat function. So if you have questions for Dr. Zamudio, please type them into the Q&A in Zoom. And following the presentation, Linnean member Jim Muchmore will lead the question and answer session. Jim is active in Hondu Herp, a nonprofit organization dedicated to safeguarding the natural lands and ecosystems of Honduras, and he's been active for quite some time with the Save the Choco project that aims to preserve biodiversity in the Choco region of Ecuador. Well, we're very happy tonight to have Dr. Kelly Zamudio with us. Kelly was born in California, and early on, her family immigrated to Peru and then to Brazil, where she was raised. She received her BA from the University of California at Berkeley and her Ph.D. from the University of Washington. She then returned to Berkeley for a post-doctoral fellowship from 1997 through 99. She began her tenure-track job at Cornell University in 2000, where she mentored and advised many undergraduates, for which she received the Stephen H. Weiss Presidential Fellowship in 2018. In 2021, Kelly joined the Department of Integrative Biology at the University of Texas at Austin, where she's actively involved in mentorship and campus-wide efforts to improve the climate for diverse students in science. Kelly's research focuses on the evolutionary processes leading to the origin of biodiversity in the highly diverse tropics. The title of her presentation tonight is Diversification and Conservation of Neotropical Amphibians. Kelly, thank you for being here tonight. We look forward to hearing your talk.
Thank you so much, Debbie. It really is wonderful to talk to you guys. I, as you heard, lived in upstate New York for many years of my life, and this time of year especially, I sometimes yearn for all those nice colors and also for the cold weather that's coming up. So if I share my video here, there we go. Let me make sure I got this. Okay, everybody see this? Okay, perfect. Thanks, Michelle. All right, it's my pleasure today to talk to you guys about diversification and conservation of neotropical amphibians. This is an area that my lab has been working on for a long time, and I'm going to basically summarize our findings in 3 different sub areas. So first, I'll tell you a little bit about stuff we've been doing in the field of sexual selection, how organisms choose each other at the time of mating, and also how that forms different mating systems in some neotropical frogs. I'm then going to switch over to talking a little bit about ecology and evolution of an infectious disease. There is a fungal epidemic out there going on. Some of you may have heard about this, which affects frogs, and that fungal epidemic is affecting some frogs more than others, and our lab is interested in understanding why that's the case. And then finally, to end on a happier note, I'm going to tell you a little bit about some work we've been doing to use eDNA, that's environmental DNA, to see if we can refine some of these lost frogs that have declined in South America. Okay, so let's get started with the sexual selection part of my talk. Okay, my lab is interested in something that we call frog reproductive modes. And just to give you a little historical context, a reproductive mode is not a single phenotype. It's actually what we call a complex trait. There are a whole bunch of things that go into what is called a reproductive mode. So people have been interested in this so-called reproductive mode since like the early natural historians, because frogs basically have a huge diversity in terms of how they reproduce. So here's a definition. Salthy and Duhlman in 1973 defined frog reproductive modes as a combination of traits that include oviposition site, ovum and clutch characteristics, rate and duration of development, stage and size of the hatchling, and the type of parental care, if any. Well, that's a mouthful, right? Like what's reproductive mode? All of that together. So it turns out that it is really all of that together. And the reason that frogs have such diversity in the way they reproduce is because of their biphasic lifestyle. It's because of how they actually use both water and land, many of them, in terms of their reproduction. So, for example, let me just give you an example. Think about egg placement, just this first column here in this graph. There are frogs across the world who put their eggs directly into water, who build an aquatic nest made out of foam. There are species across the world who put their eggs in terrestrial environments, meaning up in a tree or in a natural cavity, or they build a nest in the tree, or they build a nest in a cavity. And then there are actually frogs that keep their eggs either on or in their bodies, so very much like a mammal would, both on the male body and the female body. So that's just where you put your eggs. Now think about the fact that eggs in frogs, and majority of frogs, then turn into tadpoles, and tadpoles metamorphose into adults, right? And so on top of it, there's the place of where the tadpole develops. And you have all these different options of where tadpole develops. And then once metamorphosis happens, the little froglets can either be cared for or not by parents, and there are all these different options of how parental care can happen. So if you imagine the combination of every one of these three different columns, it becomes obvious why frogs have such reproductive diversity in terms of reproductive mode, because almost every possible combination of these three different columns exists at least in one species somewhere out there in the world. So people have known about this for a very long time. And the general idea about the evolution of frog reproductive modes has been that the more you go down on these lists, the more terrestrial you go, the more you put your eggs and your tadpoles on your body, the more you care for them, the greater independence from water you have. And people have noticed this and immediately concluded, just from these patterns, that that independence from water must have something to do with predation. It's dangerous to be in a pond. Get out of the pond, get your babies out of the pond, and maybe you would have higher reproductive fitness. So that has been the dogma for decades. There are entire generations of biologists who have studied frog reproductive modes and salamander reproductive modes, and the entire time they're interested in thinking about natural selection and how it minimizes the loss of fitness through predation if you do something special with your reproduction. Okay, get out of the water early and you reduce predation, and then all of a sudden you have a better organism that has higher fitness. All right, so that's been the sort of prevailing idea. And many of us thinking about frog reproductive modes, we're sort of like, okay, yeah, predation is important. Obviously, it's a huge issue in terms of fitness. And then in 2013, Gomez Mestre and his collaborators published this paper, and all of us working in reproductive modes were very excited about this. This was sort of the first systematic review of frog reproductive modes that was done across the globe, and also that was done phylogenetically. Okay, so they basically asked, how is it that frog reproductive modes are evolving on the phylogeny? And they found some interesting trends. First of all, they found that in the tropics, there are many more frog reproductive modes than in the temperate zones. Not a surprising result. We already knew that, but nice to see it confirmed. They then proposed that it was humidity and temperature that sort of limit the evolution of temperate terrestrial modes, meaning more in the tropics, there's more opportunity for the frogs to go terrestrial. But interestingly enough, they categorized every species that they could find data for in terms of whether the eggs were unprotected, meaning in the water, and the larvae were unprotected, and they binned all the frogs in these 4 different categories. Frogs could either be lay the eggs in the water, tadpoles in the water, or they could be protect the eggs but leave the tadpole in the water, or they could protect both the eggs and the tadpole, or they could skip eggs and tadpole. They could skip tadpole phase altogether and just go completely terrestrial, which is what happens in some frogs in the tropics. So what they found using a number of analyses on the phylogenetic tree is that there really wasn't a linear progression. It's not like, you went from an ancestral frog in the frog tree of life that was completely aquatic, eggs and tadpoles, and then all of a sudden, not all of a sudden, but or eventually ended up with the perfect frog, which had full terrestrial reproduction. That's not how it was. And what they found, and that's what these arrows show is that there was a lot of movement on the phylogeny back and forth between these reproductive phenotypes. They conclude that what this study shows is that there's the escape from predation is driving this, that multiple times on the frog tree of life, there's been a selective environment or selection to actually move out of water because of the predation selective pressure. So that's a reasonable hypothesis. But a number of my collaborators and I, who actually have worked in the tropics, both in South America and Africa, we're thinking about this. And we're like, really? Why is this all about predation? There are predators everywhere on the land and in the water. You wouldn't imagine that just getting your tadpoles and your eggs out of the water would be the only reason for this. There's a lot going on in terms of frog reproduction that has nothing to do with the predators of these particular eggs. And that's because the four of us who wrote this paper, had been thinking a lot through observations in the field and through our knowledge of behaviors during the time of reproduction, that there possibly could be a bigger role for sexual selection than had been previously than people had previously thought in this particular situation. So we thought, is it possible that sexual selection, the fact that males and competing with other males and that females actually being choosy towards males, is there something there that might be driving this change in reproductive modes across the phylogeny? And the reason we thought this is because we know from field work that there are a number of frog species that have really interesting dynamics during the time of amplexus that increase the chances of female eggs getting fertilized by more than one male. And we call this polyandry. Okay. So for example, this toad right here in the picture, I'm not sure if you can see my pointer, but there's a little vocal sac right there in the vegetation. And that is a calling male who is defending his territory. And there's a little satellite male just sitting off the side here, just waiting until female comes along to try to usurp fertilization of some of the female's eggs. We have many other examples of this. This second figure here shows a species from the Pantanal in Brazil called Leptodactylus chaquensis. This species has what we call multi-male spawning. A male builds a nest, an aquatic nest, and here in the middle, towards the top, exactly at 12 o'clock, you see a male amplexing a female. The female is releasing her eggs. The male is beating up the water and creating this foam nest. And you see all these little males all around the circle, all expressing sperm into the water, hoping that their sperm will get some increased polygyny and polyandry and get some fertilizations from that female's egg clutch. And then we have situations like this. This is a hylid tree frog from Brazil, Bocarmonoila hylax. And it has a scramble or an explosive mating system where all of a sudden at the pond in the same night, all individuals show up. They're highly synchronized. And the males end up competing with each other, grabbing and grasping females that come to the pond. And what you see here is a mating ball. This is a number of different males that are all trying to inseminate eggs that are released by this female. So you see the opportunity here for multiple males contributing to the clutch of eggs of a single female is very, very high in many of these mating situations. So to answer the questions we had, is it possible that sexual selection is playing a role in driving these particular mating systems? To test that hypothesis, that sexual selection is favoring terrestrial reproduction due to restrictions in polyandry, you have to show a couple things, right? The first you have to show is that there's a correlation between going terrestrial, getting your eggs and your tadpole out of the water, and hiding yourself actually increases your own fitness. It prevents you as a male from losing fertilizations to other males. So we decided to look at that, again, using phylogenetic methods. So we classified all the frogs in the Gomez Mestre paper as to whether their amplexus, their act of actually fertilizing eggs, the point at which males fertilize the female's eggs, was exposed, meaning it was open and other males could see what was going on, or whether it was hidden. So just as an example, this right here, Hypsiboas faber, is a hylid from Brazil. It has a terrestrial nest. It builds this nest out of the pond, but it's still exposed, right? Like other males can come, and in fact, they do come and interfere try to interfere with that male's amplexus. In contrast, Leptodactylus bufonius over here is actually quite hidden. This species actually creates this wonderful little volcano mud nest, hides inside, calls from in there, attracts a female, and not only do no other individuals fit in that little chamber, but that is where amplexus happens. Okay, so we categorized all species, did a number of different analyses to actually figure out what was driving what in terms of transitions on the phylogeny. And what we found after doing a number of different tests is that the only characters that were actually correlated on the tree were where you put your egg and where you actually have this act of amplexus, which is the point of time when males fertilize female eggs in frogs. Okay, and whether that amplexus was exposed or hidden. So what this means is that exposed or hidden amplexus is actually correlated with whether your egg is terrestrial or not. And this little diamond triangle on the right here, in this particular family of frogs, I'm using as an illustration to show you what we found. And basically what happens is that we're looking for correlations across evolutionary time. And what we can show is that when you compare terrestrial eggs, meaning when animals move their eggs onto land from water, the thickness of the arrows here on the left lower side of this diamond-shaped figure show you that that is correlated with an increase in hidden amplexus. Okay, so it's not just moving your eggs out of water onto land, it's moving your eggs out of water and at the same time moving into a situation of hidden amplexus. All right, so what that means is that this hidden amplexus is strongly correlated with terrestrial eggs, and exposed amplexus is highly correlated with aquatic eggs. Okay, so aquatic eggs tend to be mating systems. Frogs with aquatic eggs tend to have mating systems where there's more opportunity for polyandry, more competition. Whereas frogs that move their eggs terrestrially tend to have more hidden amplexus, which means less opportunity for polyandry, as we would have predicted if sexual selection might have been driving some of these transitions. So we published this in 2016 and concluded that maybe it's not all about predation, and it probably isn't, and that doesn't mean that predation isn't important. Obviously, predation is important as a selective agent, but maybe sexual selection is actually driving a lot more of this. We, during our survey, noticed that we need a lot more observations and species-specific studies to really get at some of these mechanisms in classifying all the frogs in terms of how they reproduce. There were numerous cases where we had very little data, and our main conclusion is that by focusing on one mechanism, only predation, we're basically missing out on all sorts of interesting biology that actually happens in frog reproductive biology. This panel over here on the right just shows a number of different frog reproductive modes that we ended up including in our study. So why am I telling you this story? This paper is from 2016. It's old at this point. Well, I'm telling you this because I thought it was a really interesting experience. It was like the first time that I noticed that we as collaborators actually came to a conclusion because of experiences we had had with the organisms themselves in the field, right? So the reason we sat there and thought, you know, having a beer or talking after a conference, that there could be more than this is that, you know, is that we actually had seen organisms in the field having all these different competitions, like Leptodactylus chicensis and the hylid with the mating balls. And so I think the take home here is that a lot of the diversity that we see in reproductive modes out there in frogs is probably highly underestimated. And there is so much more we could know if we had individual species specific information. So lots of work to do out there still. Okay, so that particular experience with that paper drove me into what I call like let's figure out frog reproductive modes in more detail. And we went on this search for all species that might have something interesting going on. And we've published on cannibalism and restricted breeding sites and what that has to do, what that contributes to reproductive modes. We've published on sexual selection and dimorphism, the difference in size between males and females. And I'm just going to tell you about a couple of these studies. Okay, so here's the first one. This is a study that was done by Fabio Gissá, who was my student in Brazil from Unesp, Rio Claro. And we published this in Evolution, trying to ask the question of whether male-male competition actually contributed to the differences in body size by male and females because of this mating system. This is my favorite genus of frogs, Cycloramphus. It's endemic to the Atlantic coastal forest of Brazil, and there are two flavors of Cycloramphus. One, which is terrestrial, lives on rocks along streams, mostly inhabits areas around waterfalls where the spray basically, you know, keeps them all moist. So they're the terrestrial form. And then we have Cycloramphus, which look more like this, which are entirely subterranean. Like they do come out to eat and forage, but they breed completely underground. So this was the perfect genus to test questions about the hidden versus exposed, right? Selection, because we have in the same genus species that do one thing, go underground to reproduce, and others that reproduce completely out in the open. And so the first thing we did was to build a tree for this group of frogs, and we found that, and we used that tree to reconstruct the mating system. And we found that the ancestor of all Cycloramphus back here was, in all probability, Saxiculus, the one that lives along the streams on the boulders, right? So rock living, and that have exposed mating. And that 3 times during the evolution of Cycloramphus, you have the appearance of orange on this tree. And that's the 3 times that terrestrial evolution evolved. And those are hidden or concealed amplexuses. Those species actually go onto land and build a little chamber in the ground and call for the female to join them there. So our predictions were that females should be as large as they can be. They want to have as many eggs as possible, always, right? That increases their fitness. But that this multiple evolution to concealed amplexus might actually decrease the selection for larger males. So males who are competing in streams they have to be big and strong to compete against each other. This is sexual male-male competition. Whereas if you go underground, if you go terrestrial and take your females underground, you're released from that competition, right? Because you now don't have to fight with other males. All you have to do is call the female into your underground chamber. And we reconstructed male and female size. And what we found in the same kind of analysis that I just described before is summarized down here on the right in this graphic. We found that, in fact, there is a predominance of going from stream breeding Saxiculus to going to terrestrial 3 times on the phylogenetic tree. And every time that happens, you have a transition from exposed amplexus to hidden amplexus or concealed amplexus, and that also changes the degree of dimorphism between the species. So you go from less dimorphic, meaning males and females are closer together in size, to more dimorphic in the case of terrestrial breeders. And the reason we found that that happens is because males are shrinking in size. And that as they move terrestrially, males are shrinking in size, as we predicted. And we infer that that has to do with the fact that they're released from competition with other males and don't have to be as large to compete when their amplexus is exposed. So that was pretty cool. Now, we had been thinking about mating systems as a result of sexual selection, and here's one example where we could drill down and actually look within one genus on multiple evolutions of the same thing. I'll quickly describe a study by my other student, Angele Valencia Aguilar. She's from Colombia, but is a PhD or was a PhD student at Unesp Rio Claro in Brazil, and she's now at a postdoc at Bern University. And we published this study about her work. She worked on glass frogs. Glass frogs are this really interesting and lovely group of frogs or family of frogs that are almost transparent. I'm sure some of you have actually seen them. And one of the things that distinguishes them is that they have different levels of parental care. Some species have male parental care, other species female parental care, and some species have both. The species that Angele decided to study is in the western Amazon, and it's called Hyalinobatrachium capillae. Here's a picture of one on the left, attending its 4 little clutches. This is the male. Males guard the eggs, and then they repeatedly descend down to the stream below them to hydrate, and then they bring water up and hydrate their clutches. It's been shown in previous studies that this actual active parental care and maternal care in some species actually really does increase the hatching success. So it's a real fitness benefit to individuals to do that. Okay, Angele's field site was in southwestern Amazon, and what she did during 2 seasons was monitor 36 males and their reproductive success. She visited the leaf in which they were calling every single night for the entire breeding season. And the question she was asking is, what are the determinants of female success, like of male success? Like what causes a female to choose a particular male? She was interested in all sorts of parameters. She measured everything. Perch height, leaf length. She thought maybe the leaf characteristic that the male had would matter. And the only thing that was significant after all her analyses was male attendant status. So what that means is that a male who already has a clutch, who is already attending, who is already performing parental care, gets a clutch, a second clutch, more frequently. So it's a positive feedback loop, right? The more you have parental care and show it, the more females choose to deposit eggs on your leaf. Not only was there a higher probability of getting a second clutch if you already had one, but this bottom graph shows, and male attendant status is on the x-axis here, and days in acquisition of a clutch is on the y-axis. And you can see that not only do they get more clutches, not only do males with clutches get more clutches, but they get them more quickly, more frequently than the males who aren't attending. So there seems to be something going on here in terms of the behavior of this particular mating system that is highly correlated to fitness. Right? And I'm going to skip over this real quick, but one of the things we found is that most of the clutches of the males were actually of that particular male. Like that male was the father of each of those clutches, and we determined that through genotyping. However, we did find 8 clutches that did not belong to the male who was sitting next to it. Interestingly enough, Angele has videos about this. These males who had these 8 clutches that weren't theirs did not hydrate them, did not take care of them, did not otherwise pay attention to them. And what we think might be going on here is a case of signal piracy. And what happens is that this is not impossible in a species that's this territorial and where the fitness advantages are so high if you actually reproduce. Imagine if you're a male who either fights or removes a different male, or a male gets predated and you come along and you're one of the male who's looking for a territory. Having a clutch there already present immediately increases your probability of having a second clutch and reduces the time to which you will get that second, your own clutch. So having a clutch present, even if you don't take care of it, might actually be a really good signal for females to actually consider you as a mate. Okay, so that had to do with parental care. The system's very interesting because the consequences for fitness of just this single thing, having a clutch already present on your leaf is so important. So I love these two studies because I think they show that we've come a long way in terms of thinking about how sexual selection can actually impact so highly the fitness consequences for individuals of all species, not just frogs. And it reminded me of this paper by my colleagues here at UT Austin. Mike Ryan is my colleague here, and Gil Rosenthal. They wrote this paper recently called Sexual Selection and the Ascent of Women, Mate Choice Research Since Darwin. And what they argue in this paper is that mate choice mechanisms, how individuals, both male and female, make choices about who to reproduce with, are based on all sorts of stimuli, and they're across the entire tree of life. Okay, so for example, if you're a human with a brain and you have that decision making, you have touch, feel, you have your senses, and then we also have competition and decisions being made at the level of gonads. And it doesn't matter that you're a bug or a flower or a human being, all those things are part of the tree of life and part of mate choice. Darwin, of course, did not think about this so much from the female perspective. It was part of the times, Victorian times, and Darwin focused a lot, especially in his publications, on the role of male-male competition and how that drove female choices. Females, now we know, have many more choices. And Mike and Gil write about this, and they say the trends that continue to shift away from this focus on male beauty and focus on a more holistic understanding of what males and females, or how males and females are contributing to sexual selection. And I really like this quote because at the end they say, despite a persistent structural inequity in academia, a broader range of people are studying a broader range of questions. And that means that we see a broader range of things, right? And so I think these two frog studies actually exemplify that very well. Okay, I'm gonna switch over in the last 10 minutes here because I want to leave some good time for questions to talking a little bit about the ecology and evolution of infectious disease. So many of you may have heard that there is a frog pandemic happening, and that pandemic is due to a fungal pathogen called Batrachochytrium dendrobatidis. It's a long name, so we call it BD for short. That's the name of the fungus. This fungus is particularly bad for frogs because it likes moist and aquatic environments, and frogs, of course, love moist and aquatic environments. It's a keratinophilic parasite, meaning it eats keratin, and keratin is present in the mouthparts and on the skin of frogs. And it also has a zoospore, this fungus, that lives in the water, free swimming, and then it invades the skin of the frog, and it creates these zoosporangia right here that you see in this scanning image from a scanning microscope. Those little zoosporangia breed further zoospores and then pop them out of the skin. So it's sort of a thing of like horror movies. You know, the frogs basically get infiltrated by these swimming zoospores. They create these zoosporangia. They disrupt the entire integrity of the skin of the frog. The frog ends up dying, and then all these zoospores are released into the environment to infect other frogs. Every time I talk about this, people are always asking, What's the impact of this on amphibian biodiversity, and where did this fungus come from? So let me just give you a little bit of information about that. We know that BD has really had a huge impact. We've documented at least some effect that might be BD related in 501 amphibian species over the last century. That's 6.5% of amphibians that have had some impact from this epidemic. Of those 500 and some species, 90 of them are confirmed or presumed to be extinct in the wild, and 124 are having really, really deep reductions in abundance, more than 90% population abundances. So if you look at these graphs here, as you go from blue to hotter colors to red, that's an increasing severity of decline. And so this final little brown bar over here are those species that have gone extinct. And so as you move to the right on these graphs, the situation is worse for frogs. And you can see that there are impacts all over the world, but a huge impact in Mesoamerica, in Central America, and also in South America. And we separated Brazil because many of these South American taxa over here are actually in the Andes. So we want, South America is a huge continent, so we wanted to separate them out. Okay, so there's a lot of stuff going on over here in the Americas, right? So what do we know about it? We know that most extinctions and severe declines started happening even before we knew about BD. BD was described at this arrow here in the mid-1990s, and you can see from this graph that there have been a number of declines, including some very severe ones, way before we knew about the pathogen. If you look over here at this second graph in the middle, most of the BD threatened species, the 500 I talked about, have smaller ranges. If you squint at this graph, you can see that the inner circles have more orange and red. That means that the higher disease susceptibility species are actually have smaller ranges. This is not a surprising pattern. This is common in conservation biology, that species with smaller ranges are going to be more impacted. So that is something definitely to think about. Where did it come from? I and a number of different collaborators, a global collaboration across the world, tried to build a phylogenetic tree of all the BD cultures that we had in our labs. And this is what the tree looks like. This phylogenetic tree shows a number of different branches, but let me just walk you through this. If we look at the belly button right here, the center of this circle tree, that belly button right there is the common ancestor of all the different lineages of BD in the world. And what you'll notice is that the first groups of taxa that come off, let me see if I can do this a little more colorfully here. These things right here that come off at the base of the tree, including this blue group, this pink group, and this red group, are independent of this much larger green group over here. That is what we call the global panzootic lineage. Okay, those groups there at the base of the tree are early divergent lineages that existed in various places. And then this green lineage went crazy and spread across the whole world. So if you look at this map up here above the phylogeny, you can see that the green lineage is distributed throughout the world, and that out of every continent, there is one lineage that is from that continent, and then the green lineage is spread throughout, okay? All of this evidence, not only the tree, but also this graph up here on the map shows that Asia is probably the source for this lineage of BD that went virulent and went across the entire world. All right, what are we doing with BD? So we've been doing a number of different studies, mostly focusing on the genetics and genomics of the infection process. And I'm just going to give you one little tidbit about the kinds of work that we do in this area. And that's that we actually did one study, not on frogs, but on salamanders, using eastern spotted salamanders, to ask about what the problem would be or what the consequences would be of frogs being invaded or having of salamanders having BD and then being secondarily infected by another Batrachochytrium. I'm sorry, I forgot to mention. There is BD, and there's also a second species of Batrachochytrium called B. sal. B. sal is a salamander specialist, whereas BD infects both frogs and salamanders. B. sal is also emerging out of Asia, and it has not, as far as we know, made it to the Americas yet. However, if and when B. sal arrives in the Americas, what it is going to encounter is a host community that is already infected by BD. So the reason we did this study is because we wanted to know whether double infection, co-infection, would have different consequences than being infected either by one or the other. And you might imagine that there could be two outcomes. You could have a situation where BD and B. sal both infect and then they overwhelm the host and the host dies even more quickly. Or you might imagine that BD and B. sal could co-infect the same host and they could have an antagonistic effect. They could compete with each other in the skin. They could neutralize each other, and that would decrease the disease severity. So it's important to know what's going on in terms of this co-infection process. So we did an experiment in the lab. We brought in salamanders and we did infection with BD, infection with B. sal individually, and then infection with both. And then we had some controls who were not infected at all. So let me show you quickly what we found. These are the results of a comparative gene expression. We looked at the skin and we looked at the genes that were being expressed in the skin of those particular treatment animals in the lab. And first, over here on the left, these are the results of BD versus the control. These graphs are called volcano plots. They're a way to sort of show how genes are getting expressed under a particular stress by an organism. And the reason they're called volcanoes is just because they're sort of shaped like a sideways volcano. Every single dot on this graph is a gene that is being expressed in the tissues of the animals that we collected from the experiment. If the dots are colored red, that means that they're being overexpressed in that particular treatment relative to the control. And if they're in blue, that means that they're being underexpressed, meaning the genes are being downregulated in the BD infected animals relative to the controls. So what you can see here is that when you infect a salamander with BD, it does upregulate a number of genes, all these red ones up here, and it downregulates a few, but there's not anything crazy going on. However, for B. sal, it's about the same, a little bit more upregulation, but still not overwhelming. But what we find here is that if we co-infect salamanders with both of these pathogens at the same time, not only do they upregulate a number of genes, but they downregulate a huge number of genes compared to what you would have expected given the previous comparisons. So this was our first hint that something was going on, that these two pathogens together actually have some sort of synergistic effect, and it seems to have an effect of downregulating genes. So let me skip forward here just a couple, sorry. A little too much detail, I think. Here. So what we did is we dove down deeper into exactly what those genes were all doing, the ones that were being downregulated. And what we found is that the genes that are being downregulated are these critical immune genes in the skin. And right here, if you look on the leftmost column on this graph is the expression of three different skin genes that are immune genes in the control. The next is the BD infected, the next is the B. sal, and then the final column here is the co-infected. And what you see in each of one of these three cases of these genes, indicated by the yellow arrows, is that the control always has no downregulation. You have upregulation with BD and with B. sal, but then you have downregulation with the co-infection. So what we think is going on here is that there's some sort of interaction between these two pathogens that is causing the organism to actually, to its own detriment, downregulate immune genes that should be firing off at that point, right? And that's impacting their the outcome in terms of disease. And sure enough, what my student Ana Longo found is that BD alone actually has pretty high survival. That control has pretty high survival. B-sal, which is the salamander-specific one, actually has lower survival, right? Only 40% chance of survival. But if you infect salamanders with BD and B-sal, you have the lowest probability of survival of all combinations. So this is, as Anna called it in her paper, double trouble. Something to think about in terms of the potential arrival of B-sal in the New World. Okay, I want to end the last just couple minutes talking about the rediscovery of lost frogs using environmental DNA. So I work a lot in Brazil, and Brazil has a very high amphibian diversity. Half of the species in Brazil are actually distributed in the Atlantic coastal forest. The Atlantic coastal forest is this thin forest here right along the coast, and this is its historical distribution. And as you see here on the right, those are the fragments of Atlantic forest that are left. So there's only about 16% of its original cover. It's a highly endangered biome, but it has naturally huge diversity. So since the 1970s, many amphibian biologists have been talking about declines. These are just some snippets from the literature from the 1970s, and people were talking about this back then all the time. Declines, drastic population reductions. And we now know in retrospect that at least some of these were probably associated with pathogens. So we tried to see if we could rediscover populations. Maybe there were tiny pockets that were left that could repopulate. And so we chose all these different species, and don't worry about the table, but just to show you, we chose 30 different target species at 6 different localities, and we classified these animals into 3 different categories of endangerment. They could either be declined locally, meaning we could still find them there as individuals, but the numbers in the literature were much reduced from the past. They could either be locally disappeared, meaning they were extinct or extirpated from one place, but we knew that they existed at other locations, or they could be completely extinct, disappeared. And disappeared meant the frog had not been seen in any part of its range for more than 10 years. So we went to a number of different places and used environmental DNA to try to figure out whether there were traces of the frogs still in the environment. So environmental DNA, for those of you who haven't heard about this, is actually a way of trying to capture the DNA of the organism from samples that are collected directly from the environment. Because frogs use aquatic environments, this is very convenient. We can pump and filter water in the field and bring those filters back and then go through the process of trying to assign the frog DNA that's in that filter to species, or at least to genus, depending on what we can do, compare those sequences to a reference database, which is a critical step, and that reference database then tells us, there's some trace of this frog here in the environment. So we did this at all our localities, and I'll just show you a couple results. At Estação Biológica Boracéia, which is a field station run by the University of São Paulo, we had 12 eDNA samples. We recovered 40 molecular taxonomic units, meaning things we could say were frogs. They matched frogs. Those frogs could be identified as 21 different species, but none of those species matched the 7 disappeared or endangered taxa that we were looking for. But here's a different outcome. At this park, Parque Nacional Itatiaia in Rio de Janeiro, we had 16 eDNA samples, meaning 16 filter cartridges that we brought back. We identified 20 molecular taxonomic units. Those could be attributed to 13 frog species And of those, we found 2, yay, Hylodes ornatus and Hylodes regius that were on our list of 30. This was an interesting one. This is Parque Nacional Serra da Boicana. We were looking for 3 different species there that are declined. And, we found a number of individuals, and we found one species attributed to this genus Megalosia. Unfortunately, the disappeared species we were looking for in this genus is Megalosia bocainensis. And we did not have an exact sequence because that animal is extinct, right? So we couldn't sequence it. And so we could get it to genus, and that's the only species in that genus known from this mountain range. So we inferred that this is the species we were looking for. So that was a hit to one animal that is disappeared. So in the end, after going through all localities, I want you to focus, please, on this last column here, the eDNA column. We found 7 species, all of these in green here. And those 7 species fall into 3 categories of decline. Remember the 3 categories of decline? 4 of them were considered declining. 2 of them were considered locally disappeared. And so they're locally disappeared, but potentially still there in small numbers. And then the Megalosia was the one species that we found that was has been completely not seen for the last 10 years. And we found evidence of its presence there in environmental DNA. So this was a really fun project. It is up to us to decide, obviously, whether a trace of an animal's DNA in the environment tells us anything about its chances of recovery. Clearly, those populations are not doing well, and it doesn't tell us anything about the potential for revival of the populations, but it does mean that we have one more piece of data to focus on in terms of surveys, in terms of understanding who may or may not be present and may be worthy of further conservation action. Okay, I'm going to stop right here. I hope I've convinced you that organisms are the center of everything. We're interested in traits, we're interested in the environment, and we're interested in function. And all this diversity, including reproductive diversity, comes from a combination of those things, and those also have implications for conservation. So I want to thank everybody in my lab, my collaborators, my funding sources, and thank you for listening. And I'm really happy to take any questions.
Am I here?
Yes, you are.
Can you see my,
There.
am I on the screen? All right.
Yes, that's good.
All right. Well, that was fascinating. I myself have a million questions, but let's start with some questions that have been sent in. Ralph Baker wanted to go back into the research about glass frogs. This question concerns the glass frogs that appropriate the clusters of other glass frogs. Once the frog is mate, do they hydrate their own clusters while still ignoring the other clusters?
Yeah, they do.
So they can. So he asked, do they know how to tell the difference between their cluster and the stolen clusters?
Yeah. So we don't know how they do that. We don't know the mechanism. It could be obviously chemical. It could be just recognition because that particular individual took over a leaf that already had a cluster on it, right? But she put, in the case of these 8 individuals, for at least 5 of them, she put videos on them, and it's very deliberate. Like there is no hydration of the cluster that isn't theirs. And they actually suffer, right? They have reduced hatching rates because of that.
Very interesting. Cheryl Craddock asks, what characterized species that do not appear to be affected as negatively by BD?
Sorry, I have a bunch of dinging going on here. Can you repeat that again?
Yeah. What? No problem. What characterizes species that do not appear to be affected as negatively by BD?
There are a couple correlates that we found in frogs across the world. Frogs that have relatively dry skin, so toads sometimes have a little bit more of a resistance to BD. And this is where the mating systems that I was talking about actually intersects with the second part of my talk, which is the disease part, because those diseases the mating system actually increases or decreases the exposure of frogs to different water bodies, and that is what, determines transmission. Let me see if I can turn this off. For some reason, somebody has been, sorry for the disturbance, you guys.
No worries.
Yeah, I think it may have stopped. Somebody just won something, and so everybody on this chat group is all happy about it. So, yeah, so this is where the intersection of those two things actually happens, right? So there are a number of different factors that actually affect frog susceptibility. We've also found that there are innate aspects of frogs and their immune systems that have something to do with this. Just like humans, we all vary in, resistance to different diseases because of our immune functions. And some frog species, either because of their environmental context or just because of their individual context, have better resistance to the fungus than others.
That brings me to, before the next two questions, asking you a question about BD. With my work in the tropics, I've obviously been lucky to see a lot of Atelopus species, which were heavily affected by BD. There is one population, though, the Atelopus spirelli in the western coast of Colombia in the lowlands, that for some reason has not had the effects of BD. In as much as that you can find them in the cities, in the streams right there, which is something, as you know, wildly rare.
Yeah.
And
That's a weird Atelopus though, right, Jim?
yeah. And one person who is not a scientist, who is just talking out loud, mentioned, is there any possibility that the salinity from the ocean, because you can literally find them 50 meters off the coast, has anything to do with how the disease is being affected?
Yeah, it's definitely possible. I don't know how much that species actually is affected by the salinity, but it's definitely possible. So we found Atelopus is interesting, right? So this particular genus that Jim is talking about used to have like 130 species or something like that across all of the Andes. It's endemic to the Andes. And there are a few that are sort of on the, not the highest elevation, but on the foothills. And there are a few species that go all the way down to close to lowland environments. And the crazy thing is that it's highly, highly susceptible. And the species that are, especially in the cloud forest part of the Andes, mid-elevation, where the fungus really likes to grow, good temperatures for the fungus, good humidity. So that it's been a genus that has been the highest affected. Like it's, I think 72 species have shown declines. So it's possible that this one individual species that makes it all the way down to the lowlands is actually protected by temperature. So this fungus doesn't like high temperatures. It actually thrives in higher elevations, and species that are particularly affected are the ones that are in topography and also in cooler streams and, cloud forests and so forth.
Gotcha.
So it's definitely possible.
Very cool. Sandra Beltreo is asking, for the experiment where you co-infected the frogs with BD and BSAL, did you have a co-infection control where you infected them both with BD and a different fungus and BSAL and a different fungus? I'm wondering if it mix of BD and BD-SAL specifically, or just two different fungi cause immune response and down regulation in some genes.
Yeah, that's a great question. And we're going to be following up on this study with all sorts of different combinations. We didn't do BD-SAL and another fungus and B-SAL and another fungus and BD and another fungus. That would be a really great experimental design, because it will allow us to ask about the immune function of BD relative to something more neutral, right? Not as pathogenic of frogs. Is it just an immune function because your immune is getting upregulated, or is it something specific to BD? So the answer is no, we haven't done that, but that's a really good experiment, and it's something we will be working on.
Eduardo Elias, I'm sorry, I'm dyslexic, so I'm terrible with names, would ask, can you attribute the spread of BD infection in the Neotropics and the Neotropic to the international trade and traffic of pets and amphibians?
Yeah, so in the paper that we published about the distribution of the fungus worldwide and the phylogeny, we actually talk about this. So there is some evidence that that green group, the green lineage of BD that I showed that is sort of all over the world, it is possible that that particular lineage actually spread through anthropogenic human movement, right? So this is the estimate from the phylogeny is that it was a relatively recent spread sometime in the early 1900s, 1930s, 1940s, right at the time where things were becoming globalized, transport was globalized, marine transport was widespread. So, you know, we can't say whether that's what happened. What we can say is that those other lineages, the pink, the blue, and the other one that I showed you, the red ones, are restricted to very particular regions. So one is restricted to South Africa, another one is restricted to Asia, and another one is restricted to Brazil, actually. That's one of the ones that we study. So those are what we call endemic lineages, meaning they're in lineages of pathogens that occur only in particular places. This thing, the green lineage, the global panzootic lineage, is the one that moved all over the world. And that is, we timed it in that paper to be somewhere around the early 1940s.
That brings me to another one of my questions. Are the ones that are endemic to those regions, are the amphibians affected negatively as well as the larger one, or even where you think the lineage started, did they have a mass die-off, or did those frogs co-evolve with the disease and not have as much issues?
So there have been studies about this, not from my lab, but from other labs, and it does show it. We do have evidence that there are some lineages, especially the endemic ones, that have lower virulence than this global panzootic. So the global panzootic lineage that spread all over the world has it's done all these crazy things, duplicated all these virulence genes. It's, you know, it really became a pathogen.
Wow.
So it's not very different, obviously, than COVID, right? And what we just went through, right? There are variants of viruses out there, and they're constantly tinkering with their DNA and constantly changing their genomic makeup. And then something hits, right? And that gives them fitness. And that fitness helps them spread. And that transmission rate increase helps them spread across vast places of the world. So I wouldn't expect BD to be doing anything different as a pathogen.
Interesting. Another question from Kelly Rosenheim. I can see how terrestrial systems benefit the male in terms of restricting competition, but is there a benefit for the females, enhancing female selection, perhaps?
Yes, totally. Yes. And that's why I mentioned both of those systems. So it's not just males avoiding polyandry. We talk about this in that original 2016 paper, but it's also females having control over choice, right? And so people always, I'll just give you an example. People used to always think that, you know, in species that had explosive breeding, and there are birds and frogs and all sorts of species where everybody breeds at the same time. The most famous one, for example, is, you know, the marine, you know, corals, when they put all their gametes into the water at the same time. It's like incredibly synchronous, right? And it was supposed to be a free for all, and the ideas of people, they were like, oh yeah, you know, it's completely random mating. So it turns out, even in corals, there is nothing random about it. There are interactions at the level of gametes where decisions are being made. And I say decisions because they aren't brain decisions, right? They're gamete decisions. It has to do with compatibility. It has to do with genetic makeup, and etc. So yes, the answer is yes, that there's a lot going on here that has to do also with female choice and decisions that females may have that have to do with choice of males that have to do with the fitness of their offspring.
Interesting. Sandra wanted to say, for the record, the question came from my neighbor friend, Julia Zaccello, who is an evolutionary biologist and educator, asking about the two different viruses being tried. And then she wrote, in the first study, you showed that the frog reproductive mode has a strong phylogenetic signal. Did you or could you find genes that are responsible for the shifts to the different modes?
Oh, wow, that's a good question, Sandra. It's a little tricky because it is such a complex character. Remember, I started off by saying how crazy it was that the definition, the Salty definition of reproductive modes, there's so much there. So it's a character that I'm sure we call a complex character, meaning it's not going to be dictated by one gene. So there's behavioral genes, there's, developmental genes, there's all sorts of different gene categories. And I guess I would be doubtful if I could find a limited number of genes that would explain reproductive modes. There's just too much in there, it's like it's a complex trait. It's not one thing. It's behaviors, it's where you put your egg, it's how your tadpole develop, it's whether you take care of them or not. And maybe we could find genes associated with those 4 or 5 different axes, but there's not going to be one gene for reproductive mode.
Amazing. These are great questions. Eduardo also wanted to ask, has your team looked into captive pet markets or zoo populations for the disease?
My group hasn't, but the BD community has. So the BD community worldwide has been very united in terms of really trying to figure out what's going on worldwide, which I have really appreciated. Like, we're all concerned, right? So we're all working together. And it turns out that really in the early days, back in the in the mid 2000s, 2005, 2006, we started discovering that there was diversity in BD. It wasn't all one thing, that there were these lineages, right? And that was done by a number of people who were doing genetics at the time on BD lineages. And one of the BD endemic lineages, BD Brazil, was actually discovered on a frog, a ranid frog, a bullfrog, that was in a market in Ann Arbor, Michigan, or Ypsilanti, Michigan, right outside of Ann Arbor or something. And that BD was cultivated from this captive frog that was transported from South America. In South America, they have a bunch of ranaria. It's a big thing. And that this is the product of globalization, right? That frog, we think, was exported from Uruguay from a ranarium for the meat industry to this market in Michigan, where we discovered BD Brazil on a cow. Yeah, it's crazy.
Wow.
So the pet industry is a real serious issue. And let me tell you, there's also another issue, which is BSAL, this
Right.
organism that we are thinking about in terms of its arrival here in the US. It hasn't been found in North or South America yet, as far as we know. And there are surveillance teams actually doing work to see if we can detect it. But the salamander pet industry, especially from Asia, is huge. The number of, salamanders coming out of Asia that are legally documented coming in for the pet industry is actually quite large. I've seen some estimates on the level of millions. Red-bellied newts and
it's shocking due to even just seeing what invasive plants have done to our country or countries around the world, why we're still engaging in these things.
Yeah.
And I was going to say, don't we have the largest diversity of salamanders?
Second, yeah, in Mexico and the US, we're the hotspot, the global hotspot for
So
salamanders.
it would be really bad if
Yeah.
it got here.
Yeah. Anyway, so this is, yes, the pet industry, not the zoo industry as much, but zoos have become very savvy about this because it affects their holdings as well. But they also have, the zoos have been very helpful to the community in terms of trying to learn ways to manage captive animals to make sure that it's done safely and so forth. And many zoos have been involved in BD research very actively, which is, they're part of the BD solution community. But the pet export-import industry has been a little bit challenging and will continue to be challenging, right? Until we figure out what's going on. Yeah.
All right. Well, I think that wraps up our questions, unless someone else has one last minute one, but that was really interesting. And thank you so much for taking the time to talk to us.
Yeah, thank you. And thank you for your interest. That was lovely to speak.
Yeah, thank you, Kelly. This was a great talk.
Great.
And a friend requested you on Instagram, your lab.
Okay. We'll see you there, Jim.
All right. Have a good one.
Good evening, everyone.
Bye-bye.
All right. Thank you.